Okay, so the first thing that I did was I made a cut into the sheep's sclera. That's going to be the sclera. The sclera is the outer covering of the sheep eyeball, and it's this tough, opaque sheet of connective tissue that's going to protect the inner rings of the eye and also help maintain structure and rigidity of the eyeball. The next thing, while I was cutting into my eyeball, I noticed this clear liquid coming out. And I can't really show it, but it's this sort of goopy stuff on my dissection mat. The aqueous humor, which is this clear fluid that fills the area between the lens and the cornea of the sheep eyeball, and it's majorly going to be composed of water. That's why it's so runny. And this helps maintain the shape of the eyeball, so that's very important. The next thing I'm going to show you is the cornea of the eyeball. Right here is going to be our cornea. The cornea is the transparent covering of the inner of the eye, right in the middle, and that's going to allow light to enter the eye. And on my specimen, I noticed it was like pretty cloudy when I was just looking at it, but usually it would be transparent. Let's see what else. Okay, so next I'm going to show the lens. When I was dissecting it, I thought the lens was really cool because it was just like this hard ball in the middle. But the lens is going to be this transparent structure that focuses the light coming in through the eyeball and into the cornea and into the pupil. So then after that, after that, I cut into the cornea and I removed the iris. And it's really cool to see this. So right here is going to be our iris. Right, if you can see that brownish part, that's going to be our iris. And the iris is this diaphragm that basically regulates the size of the pupil. So that's the iris in there. And then on this inner part is going to be the pupil. So that's the inner ring. And the pupil is the opening where light enters the eye. So then after that, I'd like to point out, let me get my forceps, right here on the back of the eyeball, right here, I found my optic nerve. And the optic nerve, let me put this down, the optic nerve is this bundle of nerve cells that's going to send the signals from the eye to the brain. So that's transmitting the messages of visions. So that's on the back of the eye that I just pointed out. Let's see. So then on the inner ring, I noticed this shiny reflective thing, and you can even see it on the camera. That is going to be the tapetum lucidum, and only animals have this. This isn't found in human eyes. It's this iridescent portion of the choroid tissue, and it helps with night vision by reflecting the light back into the retina, so they can see a lot better in the dark than us. And while we're here, on the back is the choroid. So this is the thin, dark sheet of the tissue behind, between the sclera and then the retina. So that is there. And then if I carefully removed some of the retina, I would also be able to see the blind spot. And the blind spot is the area of the retina where these receptor cells are going to form the optic nerve, basically. And then what else do I have to go through? What we can't see is the hyaloid fossa, and this is the indentation in the center of the vitreous body, and that would have supported the lens. And the vitreous body is the cavity between the retina and the back of the lens. What else? Rods the rods are receptor cells of the retina that are going to be responsible for perceiving lights in different intensities. What else? And oh, cones. So cones are going to be the receptor cells of the retina that are responsible for perceiving different shades of colors and such. So yeah.